In the broadest sense, the human brain works like a team. Inside it, different regions act like individual members of that team. Each one has a specific role. For example, the brain stem of this racing driver woke him up this morning, and it will guide him into sleep tonight. How does he remember the long stretch of road ahead? Memory is made possible by tissue located above the brainstem. This tissue allows him to experience strong emotions, and it allows him to remember. And covering it all is the outer layer of the brain, a wrinkled sheet of cells arranged like functional continents. At the back of the brain lies a region that relays what the eyes see. And on the surface, now at the top, a narrow ribbon relays information about touch, the feel of the wind on the driver's face, the pressure of his hand on the wheel. Just ahead of that ribbon of the brain, there is another region whose job is to orchestrate movement. It sends commands to the spinal cord, then along the arm to the hand, telling it to change gears. In front of it all is the driver's frontal lobe, the part of the brain responsible for thinking and planning. It is this bundle of tissue that provides him with a strategy to win the race. No single tissue can function without the help of the rest of the brain. The brainstem, the midbrain, and all the regions along the surface are in constant contact with each other. They are always active, even when the body sleeps. How do we know all this? Thanks, in part, to the remarkable tools that the modern-day brain detective uses to look inside the brain. The CAT scan penetrates the skull to X-ray the brain's structure, showing the precise location of actual brain tissue. Another powerful method focuses not on structure, but on activity. The PET scan captures still images that reveal which parts of the brain are working harder and which remain relatively quiet. Colour becomes information. Brightness shows intensity. Patterns show function. One of the most recent breakthroughs, however, builds on much older science. It is based on the EEG, a method that records the brain's electrical signals from sensors placed on the scalp. What makes it revolutionary is not the measurement itself, but what happens next. A computer system translates raw electrical data into a living map of the brain. What once looked like meaningless lines on paper is now transformed into colour, movement, and meaning.
Janet, now I want you to keep your eyes focused on this.
With this device, we can see the electrical changes of a brain at work. The bright colors represent positive electrical charges. Jesse Salb, the inventor, is watching the brain process a visual image. The signal from the screen flashes into the eyes and then rushes through the brain like this. Slowed down, we can watch vision deciphered. First at the back of the brain in a burst of yellow and red. About 0.3 of a second later, we can observe activity at the top of her brain as she realizes what she's just seen.
One of the most exciting recent developments in studying the functions of normal and injured brains involves combining neuroimaging techniques, joining magnetic resonance imaging, or MRI, with computer technology allows images to be reconstructed into three-dimensional pictures. One of the pioneers...
And we will now expose the brain. We see the right hemisphere on the right-hand side, left hemisphere on the left-hand side. The image will now come back to the top of the skull. The head will rotate down, now move look at the lateral surface of the brain, and then make these cuts all the way through.
Dr. Bigler's techniques are used to investigate many different pathologic states of the brain. This man had a severe brain injury from a 30-foot fall. Injured areas are shown in green. Enlargement of the internal cavities or ventricles is shown in brown. And the combined image helps researchers to understand the overall effect of the injury